Hello everyone. I'm sorry about the delay, and we tested this morning and it worked well, but it suddenly didn't work half hours ago. So before we get started, I would like to welcome you all to this SOLAS seminar. So I would like to ask everyone a favor. If you are not asking the question or making the comments, please mute yourself and during the seminar. Thank you for attending the first SOLAS seminar. SOLAS seminar series is one of SOLAS major new initiatives. And so this webinar is well registered and from more than 200 individuals and from 32 countries. And we have also students and faculties on site in Xiamen. Since this is the first seminar, so we would like to give a brief introduction to the first host, State Key Laboratory of Marine Environmental Science, Xiamen University. And then Lily will introduce briefly to the SOLAS program for those who may not familiar with SOLAS. So let's see. Xiamen University is located at the east part of China and is founded in 1921 by Mr. Tankaki. We have one of the two best ocean science programs in China, and also we have excellent key laboratory in marine science. MEL is established in 2005, and we have more than 210 staffs, including about 97 faculty members who are working on the marine biogeochemistry and ecosystem dynamics, and also the related research topics. Within last 2 decades, MEL has built a comprehensive capability for the ocean observation and also for the marine-related research. Looking at this PPT, we have marine stations and have a Tankaki research vessel, and up in the air we have satellites, 2 satellites for the remote sensing study, and we also have other buoys mooring system over the ocean. We have some fellowship, a variety of a set of variety for the fellowship, including visiting fellowship for distinguished senior and early career scientists. MEL also offer postdoc fellowship and the PhD fellowship. At the same time, MEL has a summer research fellowship for undergrads. So for these who are interested in collaborating and visiting MEL, you can go and visit our website or contact us for further details. Yeah. And last slide, MEL has host international conference, so-called Xiamen Symposium on Marine Environmental Science. The next one, it's held for every 2 years. The next one will be in Xiamen next January, that is 2023, January 9th to 12th. Yeah. So we are welcome all of you to join us on site or virtually online. without further introduction, I would like to invite Dr. Lee to give a brief introduction on the SOLAS program.
So, all right. So before I start, on behalf of the SOLAS Scientific Steering Committee and the International Project Office, I would like to thank Bimbing and Mel for hosting the very first seminar of the SOLAS Seminar series. And I see many familiar faces here and also many familiar names online, actually. So welcome, both old friends and newcomers. And I would like to mention that Ming Han, SOLAS co-chairs of SSC, he does want to join us today, but couldn't make it because of unexpected reasons. So please don't sue us for false advertisement. All right, let's start. So SOLAS stands for Surface Ocean Lower Atmosphere Study. It is a global research program launched nearly 2 decades ago and dedicating to studying the interactions between ocean and the atmosphere. Under the sponsorship of Future Earth, the International Commission on Atmospheric Chemistry and Global Pollution, the Scientific Committee on Oceanic Research, and the World Climate Research Program. The mission of SOLAS is to quantify the biogeochemical and physical processes between the ocean and atmosphere, and to understand how this aerosols-coupled system affects and is affected by the climate and global change. The second phase of the SOLAS program is organized around 5 internally connected core themes, which are greenhouse gases and the oceans, air-sea interface and fluxes of mass and energy, atmospheric deposition and the ocean biogeochemistry, interconnections between aerosols, clouds, and marine ecosystem, which is the topic of today's seminar, and ocean biogeochemical control on atmospheric chemistry. And we identified a number of particularly important cross-cutting themes that integrate all 5 core themes, such as integrated topics on vulnerable ecosystems. And we have an initiative evaluating the environmental efficacy and impacts of climate intervention. For the solar science and the society theme, we are looking at how air-sea interaction science can contribute to policy development, as well as how policy and human activities impacts this air-sea coupled system. And the Scientific Steering Committee is responsible for providing scientific guidance to the development and implementation of the SOLAS program, which is currently consists of 17 members from 14 countries. And we are currently calling the applications to the SSC, which will be closed by May the 20th. So if you would like to submit a nomination or self-nomination, please scan the QR code on the screen for more details. And the international project offices of the SOLAS program are hosted by MEL, and another one is hosted by the National University of Ireland, Galway. So together with my colleagues in Ireland, we together, that's me, by the way, Photoshop, of course. So we together facilitate the integration and the synthesis of international activities and research across multiple levels. SOLAS has scientific networks in 31 countries and regions with 36 representatives who report relevant scientific progress of their countries and regions to SOLAS on an annual basis. And we sponsor and endorse national, regional, or international research projects and time series stations across the globe. For example, we co-sponsor this integrated ocean carbon research, which is currently under the auspices of IOC UNESCO. And we sponsor the SOLAS Inward Ocean Acidification Working Group, which is part of the advisory board of the IEAE Ocean Acidification International Coordination Center. And we also co-sponsor 2 polar relevant projects, which are BACSI and CATCH. So we have been dedicating to integrating the ocean and atmospheric research to ensure effective implementation of science in policy and climate and the decision-making on climate and environmental health. For example, via the UN Decade of Ocean Science for Sustainable Development, as well as the UN Framework Convention on Climate Change relevant activities. So in addition to over 100 workshops that has been organized, there are two major SOLAS event series. One is the SOLAS Summer School, which has been a great success. According to a recent survey, most of the alumni remain in academia, and some of them have becoming world-leading scientists. And the next in-person SOLAS Summer School will be held next summer in Cape Verde. And the other SOLAS major event is the Open Science Conference, and the 8th one is being organized as a hybrid event in Cape Town, South Africa this September. And please know that the abstract submission and the registration will be open very soon. And the third one of the SOLAS major events is the SOLAS Seminar Series, which is exactly launched officially today. So hope every registrants and participants of the event today will enjoy it. And here are some more SOLAS upcoming activities. And please join our mailing list or follow us on Twitter to be kept up to date. Thank you.
So today we have 3 invite talks. Each talk will be about 30 minutes, including Q&A. We will have a short discussion after the talks. And if you have questions, you also can save that for later. And you can also type it in the chat box in the Zoom system. And so I will find that later for the speakers. Our first speaker is Patricia Quinn. Dr. Quinn, could you please share your slides while I introduce you?
Okay.
Okay, thank you. Dr. Quinn is a research chemist at NOAA Pacific Marine Environmental Laboratory since 1993. She got her PhD degree from University of Washington in 1988. She published numerous papers and many awards. Dr. Quinn was elected as the HU Fellow 2010 and then AAAS Fellow in 2019. She also served as SOLAS SSC members from 2008 to 2014. Today, Dr. Quinn will talk about the connections between marine ecosystem and atmospheric aerosol and the clouds. Dr. Quinn, please.
So can you see the big presentation?
Yes.
No,
It's
no,
on
no, no,
speaker mode.
Yeah, it's not a big.
Okay, let me swap displays and see if that
Yeah.
helps. How about now?
Yes, good. We can
Okay.
see that.
Okay, thank you. All right, well, first of all, thank you, organizing committee, for inviting me to speak at this first SOLAS Science Seminar. So I'm going to talk about, as was said, connections between marine ecosystems and atmospheric aerosols and clouds. I'm trying to advance my slide. Okay. Okay, so why focus on marine ecosystems and their impacts on aerosols and clouds? Well, clouds and aerosols continue to contribute the largest uncertainty to estimates of the Earth's changing energy budget, and uncertainties in emissions of natural aerosols, including sea spray and biogenic sulfate, account for about half of the variance in aerosol forcing since 1750. So reducing uncertainties in climate forcing estimates requires a better understanding of the role of natural aerosols. So I'm going to talk about today is the sources of CCN, or cloud condensation nuclei, to the remote marine atmosphere, specifically sea spray and non-sea salt sulfate. I'm going to talk about the North Atlantic Aerosol and Marine Ecosystem Study, or NAMES, the measurement methods that were used in this analysis, and then the results of the series of experiments. So clouds do not form without aerosol particles, and aerosol particles that nucleate to form cloud droplets are called cloud condensation nuclei, or CCN, and is shown by this satellite image of ship tracks off the coast of the Western US. You can see that the particles are impacting the cloud reflectivity or albedo, and they also impact lifetime and extent of the clouds. So particles that are sufficiently large and water soluble take up water vapor and become cloud condensation nuclei, or CCN, and then they can grow or activate to form cloud droplets. So how exactly do we determine sufficiently large and water soluble? Well, we do that based on Cooler theory. And on the left is a graph I'm not going to go into too much detail, but it is water vapor supersaturation on the y-axis versus droplet diameter on the x-axis. And it shows three different curves, which are for three different sizes of ammonium sulfate aerosol. So at this low end of the size range, about tens of nanometers in diameter, the Raoult effect comes into play, so that particles that contain some water-soluble material form an aqueous droplet when water condenses onto them. This lowers the vapor pressure over the droplet, which leads them to take up more water and grow. And then the Kelvin effect comes into play as on the larger particles, because larger particles act more like a flat surface than a curved surface. And this lowers the vapor pressure over the droplet. So big drops can take up more water than little drops. But once a droplet takes enough water up to reach its critical diameter, which in the plot on the left is the maximum in each of those curves, for a given supersaturation, it can then activate and grow into a cloud droplet. And that critical diameter depends on the initial dry particle size and composition. And every particle type has a unique Kohler curve. And I'll be coming back to this later in the talk, because this is how we do some of our CCN calculations. So the first source of CCN to the remote marine atmosphere I'm going to talk about is sea spray aerosol. And that little cartoon shows you the process of wind-driven production of sea spray aerosol. So breaking waves entrain air bubbles into ocean surface waters. Those bubbles then scavenge organic matter as they rise to the surface. And when they burst at the surface, they release sea spray particles that are composed of inorganic salts and the scavenged organic matter. So this just shows many of the organic carbon components in seawater that could get scavenged through this bubble bursting process. And it's as a function of size. So on the left in the blue is dissolved organic carbon, or DOC, and that's operationally defined as what passes through a 0.2 micron filter. And it contains viruses, macromolecules, microgels, and colloids. And that makes up about 90% of the seawater organic carbon pool. And then the right in the green shows a particulate organic carbon, or POC, and that is what is retained on a 0.2 micron filter. And that's zooplankton, phytoplankton, bacteria, macrogels. And that makes up about 10% of the seawater organic carbon pool. And so this next slide shows what the composition of sea spray aerosol particles looks like. And this is sea spray aerosol particles based on aerosol that's generated in the Scripps wave tank. So it's going from 30 nanometers up to about 3 microns. And you can see on the left side of the plot that most of the smaller particles are composed of organics. So these are those organics that are scavenged in the bubble bursting process. And the ones that were measured here are amino acids, fatty acids, lipids. And then there's this middle range where you have these internally mixed particles that are composed of both sea salt and organics. And then the larger particles are primarily sea salt as detected by sodium, chloride, magnesium, and potassium. And at least in this case, as generated in this wave tank, there were some larger biological particles, primarily bacterial cells. So the second source of CCN to the marine atmosphere I'm going to talk about is non-sea salt sulfate. And this is a very complicated diagram. The bottom half under the sea surface is showing the ocean sulfur cycle. So plankton release dimethyl sulfonium propionate, or DMSP, and then that DMSP is enzymatically cleaved to form dimethyl sulfide, or DMS. And the DMS is then emitted to the atmosphere and oxidized to form non-sea salt sulfate and methane sulfonic acid. And that sulfate then can become a CCM. But I also don't want it to go without saying that there is now a newly found oxidation product of DMS in the marine atmosphere, and that's the upper right-hand molecule HPMTF. And I'm not going to say any more about it, but just to know that the irreversible loss of this molecule in clouds has implications for the role of DMS in new particle production and particle growth. So this is just putting together the sources of CCN to the remote marine atmosphere. It's dimethyl sulfide, which is coming from phytoplankton in the ocean. It gets oxidized in the atmosphere to form sulfate, and the sulfate then can become CCN. And it's also the bubble bursting at the ocean surface, emitting sea salt plus organics, and those two then can become cloud condensation nuclei. So the potential changes to the marine ecosystem that could impact CCN in clouds include an increased concentration of dimethyl sulfide, leading to increased concentrations of non-sea salt sulfate aerosol and CCN. And then also there could potentially be changes in the composition or concentration of seawater organic matter that could impact aerosol chemical composition and CCN activity, and that would either increase or decrease CCN concentrations. So the Western North Atlantic is a very good place to start looking for linkages between marine ecosystems, aerosols, cloud condensation nuclei, and clouds. This chart just shows a satellite image of chlorophyll in the North Atlantic with Greenland on the left and Norway on the right. This is the largest annual recurring phytoplankton bloom in the global ocean. There's a very large spatial and season variability in plankton biomass and composition, and periods of low aerosol concentrations during remote marine conditions provide opportunities to detect impacts of the bloom on aerosol properties and CCN concentrations. So this is just showing a cartoon view of the annual plankton cycle in the North Atlantic. The y-axis is ocean depth, and this is the ocean seawater mixed layer depth shown by the curve. And then it's just progressing across the seasons. So if you focus on the middle during the winter transition of the bloom, you get higher wind speeds. So this deepens the mixed layer. And then as you go towards early spring, there's a shoaling or the shallowing of the mixed layer, an increase in sunlight, and then increasing phytoplankton concentrations. And that leads to the bloom climax. And then late spring and early summer, you have a depletion of nutrients. The phytoplankton are consumed, more DMSP is released, and you end up with higher seawater and atmospheric DMS concentrations. So just how this relates to our two sources of CCN to the marine atmosphere. One would expect that in winter, where you have the highest wind speeds, you'd have more emission of sea spray aerosol. And then during the late spring, early summer, with the highest DMS emissions, you would expect to have more sulfate aerosol. So there were this series of experiments that took place that focused on the North Atlantic phytoplankton bloom. And they were 5 different cruises designed to target seasonality of the plankton bloom. And the plankton community composition and concentration, physiological stress, and chlorophyll varied widely across these different cruises. So going back to the plankton cycle, just putting into context the different cruises that were used to investigate it. NAMES 3 took place in September, so that was the declining phase of the bloom. NAMES 1 took place in November, so that was the winter transition of the bloom with wind speeds at a maximum. NAMES 4 took place in March, so that was a period of phytoplankton accumulation. And then WAX 2, which was also in the same region and the same season as NAMES 2, was late summer, early spring when DMS was at a maximum. So this is talking about the measurement methods that we used. First of all, we took all of these cruises and we selected time periods during the cruise that were the most remote marine. And we did that to look at the impacts of the bloom and to minimize continental aerosol sources. So we took calculated air mass back trajectories. So on the right is shown an air mass back trajectory for a day in November, but it just shows a typical trajectory coming down from the Arctic across northern Canada into the ship. So that's very clean marine air. We also looked at radon, which is a tracer of continental air. So we used low radon concentrations, low particle number concentrations, and low aerosol light absorption coefficients, which is a proxy for black carbon or soot. So taking all of those combined factors, we pulled out periods that were typical of remote marine conditions. And then we used two approaches for assessing sources of CCN to the marine atmosphere. The first approach was we used the measured aerosol chemical composition and CCN concentrations. So we measured the chemical composition using size segregated aerosol collection with impactors, and one of the impactors is shown on the right there. That's actually sea salt deposited in small little jet plate drops. And then we analyzed the samples by ion chromatography for non-sea salt sulfate and sea salt. We also did thermal optical analysis for organic carbon, and we had a DMT CCN counter for CCN concentrations. So we compared non-sea salt sulfate and sea salt mass concentrations to measured CCN concentrations. We also took particle number size distributions covering the range of 20 nanometers to 8 microns. And we did a log normal fit to calculate modal number concentrations. So what I mean by that is we looked at the accumulation mode, which in this case was primarily non-sea salt sulfate. And then we also looked at the coarse mode, which was sea salt. And then we used those modal CCN number concentrations and Cooler theory. I mean, we use the Cooler theory to calculate those modal CCN number concentrations. So this is the first result. This is a comparison of the measured chemical composition and the measured CCN concentrations. So it's CCN concentration on the y-axis, and then the first plot on the left is sulfate, and the middle plot is particulate organic matter, and the rightmost plot is sea spray aerosol. And this is if we combine all of the results from all of the different cruises, we only see a significant correlation between sulfate and CCN concentration. So that indicates that sulfate is primarily forming the CCN over the western North Atlantic. But if we pull out just data from the wintertime cruise, which is shown in red, so that's NAMES 1, we do get a pretty good correlation between sea spray aerosol and CCN. So sea spray aerosol is well correlated with CCN in November when wind speeds are high and the plankton and DMS concentrations were low. So this is the second approach I mentioned. This is taking a log normal fit of remote marine particle number size distributions to determine the number of particles in each of these modes. So this is for both plots, it's number concentration on the y-axis versus diameter on the x-axis. And the only difference is the plot on the left is a linear y-axis, and the plot on the right is a logarithmic y-axis. And that's just so to help you visualize the sea spray aerosol mode a little better. So the circles are what was measured. And then the different colors represent the log normal fits. So the green is an Aiken mode, which is basically nucleation of gas phase species to form new particles. Red shows the accumulation mode, and it's called that because mass is accumulated as particles age in the atmosphere, and that in this case is mainly sulfate. And then the sea spray aerosol mode is shown in blue. So that's the wind-driven production of sea spray. So this shows the total measured particle number concentration for each season. So that's total number on the y-axis, and then it goes from November to March, May, June, and September. And this is if you look at the size distribution plot on the right, it's just taking the sum that's shown there in black. So it's all the particles covering every mode. And it's clear that the lowest particle number concentrations were observed in November. I mean, that's like less than 100 per cubic centimeter. And then the March, May, June, September number concentrations are all pretty similar. So then we can calculate the number fraction for each mode and season. So this is just taking the number concentration we calculate from the log normal fit in each mode and dividing by the total number concentration. And again, it's for the different seasons. So the Aiken mode is in black, the accumulation mode is in red, and the sea spray mode is in blue. So during winter, which is the leftmost plot, November, the sea spray aerosol mode makes up about 30% of the total particle number concentration. But in the other season, it makes up less than 10%. So sea spray aerosol number concentrations are low, which limits their contribution to the marine CCM population. And instead, it's sulfate that's dominating the particle number concentration in all seasons. So now we can take the cooler theory that I talked about initially, our measured chemical composition and our measured size distributions, and we can estimate the contribution of sulfate and sea spray to the marine atmosphere CCM population. So this top plot is the number fraction of CCN that's made up of either the biogenic sulfate aerosol or sea spray aerosol, again with each season. So sulfate makes up the majority of the CCN in March, May, June, and September. And the sea salt is minimal in those seasons, but for November, when atmospheric DMS concentrations were at a minimum and the bloom accumulation had not yet occurred, the wind speed was lofting more sea spray into the atmosphere, so that sea spray aerosol actually dominated the CCN concentration. And the bottom plot shows the same seasonal averages, but it's wind speed in blue and then atmospheric dimethyl sulfide in red. So you can see for names 1 in November, the wind speeds were the highest, and that correlates with the highest sea spray number fraction of CCN. And then in May and June, when atmospheric DMS concentrations were highest, there was the highest sulfate number fraction of CCN. So that's just talking about the Western North Atlantic. So what do other cruises in remote marine regions reveal about sources of CCN to the marine atmosphere? So we took, we've been on many cruises, and we took the ones that were most remote marine in nature. We used the Kohler curve relationship and measured composition and size distributions to calculate the fraction of CCN that were non-sea salt sulfate versus sea spray. And then we bend the data into latitude bands. And this is the result. So the x-axis is latitude going from 80 south to 80 north. And the y-axis percent supersaturation in the CCN instrument. And the top shows the CCN number fraction due to sea spray. So sea spray contributed up to 30% of CCN in the high southern latitudes, but was negligible elsewhere. The middle plot shows biogenic sulfate in that Aiken mode that contains more freshly formed particles. So the biogenic sulfate in this mode contributed to CCN in the mid latitudes This was mostly due to entrainment from the free troposphere, but this contribution was only significant at high supersaturations due to the small particle size. And these supersaturations are probably unrealistic for the real atmosphere. And then the bottom plot shows this contribution to the CCN number fraction due to biogenic sulfate in the accumulation mode. So in every ocean region, with the exception of the high southern latitudes, sulfate aerosol dominates the CCN population. So to conclude, observations during NAMES and over other global oceans indicate that biogenic sulfate provides the primary connection between marine ecosystems and aerosol and cloud properties. And as a result, there is a need to better understand the mechanisms that control the burden of sulfate in the marine boundary layer in order to reduce uncertainties in climate simulations. Thank you.
Thank you, Dr. Quinn. Do you have any questions from the virtual participants? Okay, let me take a liberty to ask one question. So while I was thinking about the questions, so in your presentation, you briefly mentioned that the DMA's lifetime in the atmosphere. So in the past few decades, so the cloud hypothesis has been tested and developed. So my question is, how do you see it in developed over the decades, and what adds the complexities into it?
I guess you're asking what I feel about the cloud hypothesis at this point?
Yes.
Yes. I think that the cloud hypothesis was useful because it brought together people looking at the problem from different angles, but I think that there's too many difficulties going from that emission of DMS from that particular phytoplankton bloom, and then having that turn into sulfate, and then that feeding back onto the same phytoplankton bloom. So I just don't think the mechanism is in place for it to have a climate regulation role.
Okay. Now we have one question from the onset.
Hi, Dr. Quinn. This is Jiaxue, speaker. I have one question about your presentation. As the method path shows, the non-sisal sulfate is in the accumulation mode, and the sisal sulfate is and the sea salt is in the coast mode. If the MSA or sulfate acid is absorbed on the surface of sea salt due to the acid-base adsorption, does non-sisal sulfate contain this kind of particles, and how to qualify this kind of particles?
I'm sorry, this kind of what? I don't understand the end of the question.
The last part of the question.
How to say? I mean the MSA or sulfate acid will absorb on the surface of the sea salt due
Right.
to how to does the non-sisal sulfate contain this kind of particles, and how to qualify this kind of particles?
I'm still not understanding. I think you're asking if the difference between where sulfate ends up in the aerosol and where methane sulfonic acid ends up in the aerosol. So sulfate ends up in the accumulation mode because it's going through cloud cycles and glomming onto these smaller particles whereas methane sulfonic acid there is some submicron, but it does end up in the coarse mode because it's coagulating onto basic sea salt particles.
So we have two questions on the chat box. So the first one is, to what extent the marine VOCs other than DMS could influence the number of the CCNs?
That's a good question. And I don't think we know the answer, but I think that we do know that the other VOCs are much lower in concentration, at least what we know for now are much lower in concentration than the concentrations of DMS coming out of the ocean. So I think DMS is the largest source of VOCs coming out of the ocean.
Okay, thank you. So the other one is, did you have a chance to look at the nucleation mode aerosols? Any hints on the source of the nucleation mode aerosols?
Yeah, I think that in the marine atmosphere, the nucleation mode aerosols are coming from the upper troposphere. I think that dimethyl sulfide gets lofted into the upper troposphere and it's formed in these cloud outflow regions, and then that gets subsides back into the boundary layer. So I don't think that there's very limited evidence of nucleation happening in the boundary layer.
Okay, I guess I have the similar question to San Diego's. So are these considerations for DMS drop formation valid also in ice clouds? Yeah. So we haven't touched any of the ice nucleation part yet for the DMS oxidation products. Yeah? And so that's the question. Yeah.
Yeah, I don't know the answer to that. I mean, they're not widely spoken of as material for ice nucleation, but I don't know the answer to that.
Okay, so yeah, in our group, we also did some work on the ice nucleation for these particles that derived from the DMS oxidation. So yeah, maybe next time we can talk about the ice nucleation from all these marine-related DMS productions. There's another question. I see low DMS but high sulfate. Could you give some explanations on these differences?
So I think this sort of goes back into the claw question too. I mean, just because you have low DMS, it doesn't impact all of the sulfate that's there at the same spot in the ocean. So the sulfate could have come from somewhere else, come down from the free troposphere and ended up in the boundary layer. So I think there's a disconnect between DMS emission and the sulfate that you are measuring at that same spot.
Mm-hmm. Okay, thank you. Jen Wang raised the hand, so please, Jen Wang.
Hi, Trish. I just want to have a clarification question. So you calculate the CCN. I think the sea salt contribution is from the fitted coarse mode. And then the sulfate contribution is based on assuming all other species are basically contributing to the accumulation mode, and then you based on the composition. So I think it's very comprehensive analysis about different regions about the contribution from sea salt. I want to ask you about the question. You're probably aware there's a recent paper talking about derive the sea salt particles in a slightly different way using the hygroscopicity instead of fitting, and based on one particular location that they found, there appears to be that will give somewhat higher contribution from sea salt when compared to derive the sea salt contribution from the feeding the mode. I just want to see what are your comments or any thoughts on whether that is more widespread in different regions, or what that would potentially modify the numbers, or
I just can't believe it's going to modify the numbers enough
okay.
to make enough of a difference. But yeah, that's a good question.
Okay, great. So we'll have a nice discussion, and we still have time later for the general discussion. So thank you, Dr. Clay, and could you please stop the sharing, and Jianwang, please share your slides.
Okay, so
Okay. Now the second speaker is Jianwang from Washington University in St. Louis. He is now a professor in the department. He was a scientist in Brookhaven National Laboratory before moving to St. Louis. He got his PhD degree from Caltech in 2003. Dr. Wang won the Kenneth Whitby Award from AAR in 2013, and then NASA Group Achievement Award in 2021. So Dr. Wang will give us the talk on the aerosol properties and the processes in the Eastern North Atlantic. Dr. Wang, please.
okay, so can everybody see the shared screen and also hear me? Okay.
Yes.
Okay. All right. So I guess we will first thank you for the organizer for the opportunity to give this presentation. I'm really happy to share some of the recent work my group have done. And today I'll be focusing on the aerosol property and process in slightly different location, but it's very close. It's in Eastern North Atlantic. Let's see. First, it's not advancing the slide. Okay, so the motivation is, as Trish also gave a very nice introduction about cloud formation, the aerosol serving as the seasonal cloud droplets. We know the marine low clouds have very extensive coverage, and they're very sensitive to changing aerosol population. And showing here is the aerosol first indirect forcing, simulated first indirect forcing, and also associated uncertainties. And you can see those forcings and uncertainties are dominated by marine low-level clouds, essentially clouds formed in the marine boundary layer, particularly in the mid-latitude regions, such as in Eastern North Atlantic. And one of the major contributions to this uncertainty is due to our incomplete understanding of aerosol property and processes in the marine boundary layer. If you look at Eastern North Atlantic, the aerosols there can have a variety of different sources, both natural and anthropogenic. So it is a quite ideal region to study aerosol processes in a remote marine environment that is periodically perturbed by anthropogenic emission, for example, from North America. So I also, I was able, with the courtesy of Trish, I got this figure, nice figure, and Trish already explained some of the very important processes. So if you look at the sources, they have a sea spray aerosol, as Trish have talked about. And today I'll be talking, mostly focus on some of the other processes. You know, the aerosols can also entrain from free troposphere into the marine boundary layer. And in the free troposphere, we have aerosols that are transported, long-range transportable continental emissions, but also new particles that formed from the DMS oxidation products, such as sulfuric acid. And once they're entrained into the boundary layer, they can undergo a variety of processes, such as condensation, coagulation, and cloud processing. And I also want to point out, we often neglect a very important process that's actually removal, which is also a big lever in determining the aerosol concentration. In the marine boundary layer, the most significant process on removing cesium is actually through coalescence scavenging. But I will not focusing on that today. So I'll be using measurements in a recent field campaign called Aerosol and Cloud Experiments in Eastern North Atlantic. And this campaign includes a long-term measurement at a site on Graciosa Island from June 2017 to August 2018. And if you see here, this gives you, essentially indicating the general geographic area of the Azores. And it consists of a group of islands. The Graciosa Island is here, and the measurement site is very close to the North Shore. In addition to the long-term, relatively long-term measurement on the ground, We also have aircraft measurements during 2 seasons, one during the summer, one during the winter. Altogether, we had about 40 flights. And those lines represent the flat tracks, give you a general idea about where we were sampling, and mostly around in the Azores region. If you look at the size distributions, and I will be always using the red for the summertime and the blue for the wintertime. And in the marine boundary layer, we see this typical bimodal size distributions, and with accumulation mode, sizes are about the same for both seasons. But for the winter time, the Aitken mode sizes are somewhat smaller, 20 nanometers smaller than the summertime. But what's quite interesting is if you look at the aerosol size distribution during this winter time, we have the larger fraction with the particles between below 20 nanometer in the marine boundary layer, both in marine boundary layer and in the lower free troposphere. So the traditional, we think that those small particles are typically produced in the free troposphere. So the new nucleation mode particles observed in the marine boundary are typically attributed to the new particles formed in the free troposphere and entrained into the boundary layer. So, but if that's the case, we would expect we should have much lower concentration of those small particles in the marine boundary layer, which is not what we have seen in this particular campaign. And so it turns out we found actually the new particles actually can form in the marine boundary layer actually quite regularly following the passage of cold front. And so I'm giving an example. This is February 14th on 2018. We have a cold front passing through the Azores, and you can see this band of precipitation which indicating the cold front. And on February 15th and 16th, you can see the Azores was behind the cold front. And as you have the cold air advect over the warm water, the boundary layer become deeper, and also they become decoupled. What I mean decoupled is that you have the upper part of the boundary layer no longer well mixed with the lower part. They're no longer being well mixed. Under those conditions, the cloud tends to often forms open cell convection. So you can see here the cell. So you see the clouds along the edge of the cell, while the center of the cell is clear and cloud free. So these are the measurements on board the research aircraft on 16th. Essentially, this is the day after the passage of cold front. And if you look at the vertical profile, potential temperature, water vapor mixing ratio, and also the red is CO, you can see we have a pretty deep boundary layer and also decoupled. The reason we say they're decoupled is we have the water vapor mixing ratio is much lower, is substantially lower compared to the near surface. So they're not efficiently mixed, but CO concentration is still uniform within this boundary layer. And the black line represents the liquid water content. So this gives us essentially indicating the cloud will form as the upper part of the marine boundary layer. And M over Z63 essentially represents the DMS. We see there's enhanced concentration of DMS in the boundary layer. This obviously is due to the surface emission from the ocean surface. The blue line here, this represents the ratio of the concentration of particles larger than 3 nanometers over the concentration particle larger than 10 nanometers. So if we don't have any newly formed particles between 3 and 10 nanometers, the two concentration would be the same, so you get a ratio of 1. So when we see a ratio that is substantially above 1, that would indicating the presence of newly formed particles and or recent new particle formation events. And you can see on that day, at the upper part of the marine boundary layer, we see this enhanced the concentration ratio, this indicating recent new particle formation. This is also supported by the measurement of size distribution at this level, which is showing the concentration keep increasing as the particle size is decreasing towards the 10 nanometers. We also see this anti-correlation between the ratio of the concentration and the surface area. This is also consistent in our picture about new particle formation, because new particle formation typically occurs when you have reduced surface area, which can scavenging, nucleating, or condensation species. I'm showing here, this is a flat track that is colored by the ratio. And you can see we have this enhanced ratio over a distance about 50 kilometers, which indicating this is a more of a regional instead of isolated events. So if you look at the ratio in the lower part of the marine boundary layer, it's obviously much lower than the upper part, but still is substantially statistically above 1. This indicates that newly formed particles were transported to the lower level. And the side distribution near the ocean surface shows we have slightly increased nucleation mode diameter, indicating those particles were underwent growth as they were transported to near ocean surface. So we see a number of those cases, and essentially all of those cases are behind essentially following the passage cold front. So the conceptual picture is when you have, after the passage cold front, you have this deeper boundary layer, which is showing, which essentially have open cell convection of the cloud. And the cloud along the edges will be able to efficiently transport those cumulus clouds. Along the edges can efficiently transport reactive gases such as DMS into the upper level. And we also have strong removal by drizzle inside the clouds. So this leads to reduced surface area in the upper decoupled layer. So if you look at the clean regions between the clouds, we have enhanced reactive gas concentration, lower surface area. There's also increased actinic fluxes due to reflection by the side of clouds. And also we have a lower temperature after the passage cold front. So all of these are very favorable conditions for new particle formation to occur. And once those new particles are formed, they could be transported through turbulent mixing across the boundary to near the ocean surface where they can be observed near the ground. So we had this 1 year sort of a continuous measurement. So we identified 67 events, growth events, and during which we see these nucleation mode particles shows sort of more or less a continuous growth to larger size. We attribute those nucleation mode particles to what have formed in the upper part marine boundary layer, as we've shown from aircraft measurement. And all those cases were identified following the passage of cold front. As you can see, this is the precipitation associated with cold front, and then we see this nucleation mode start to grow. So I was showing some of the measurements from the ACNA measurements. We also look at the measurement from NAMES campaign. This is a campaign Trish just talked about a moment ago. We here I'm focusing more on the aircraft measurement. Essentially, we have aircraft measurement during 3 campaigns, the 3 seasons, November, May, and September. And what we have done is we essentially compare the aerosol surface area, temperature, DMS concentration, and relative humidity for the time when we observe new particle, newly formed particles, which is indicated in red, and also when the new particles are absent. Essentially, there's outside new particle formation events. And you can see whenever we see this new particle formations in the boundary layer, we see there's a reduced surface area, lower temperature, and also increased DMS. This is essentially consistent with what we have found from the data collected from ACNA. That is consistent with this new particle formation following the passage of cold front. And we also, one of the advantage with NAMES is the aircraft flying to much higher altitude. Unlike during the ACNA, the airplane mainly stayed below 3,000 meters. So we can also look into the new particle formation in the free troposphere. And you can see, we see new particle formation occurs under the condition we have a much lower surface area, elevated DMS, and also high relative humidity. This is consistent with the new particles are formed in the outflow of a convective cloud. This is a traditional picture that how new particles occur, where new particle formation occurs over the open ocean. So now we're comparing the particle number concentration. This is newly formed particle concentration, and this is the concentration sort of in the nucleation / Aitken mode particles, and those have a relatively longer lifetime, so they can grow and reach a CCN size. And you can see we have this enhanced newly formed particles and Aitken mode nucleation / Aitken mode particles during those new particle formation events. which are substantially higher than in the free troposphere, indicating this might serve as important sources of the CCN in the marine boundary layer. We also looked into the frequency of new particle formation. This is defined as the time period when we observe new particles to the total sampling time of the flight for that altitude range. And you can see we have this local maximum of frequency about 6% at the top of the marine boundary layer. So this is still less frequent than what's typically observed in the free troposphere in outflow, which is about 15%. So now I want to come back to the eastern North Atlantic. Now, again, this is a measurement on the G1 aircraft during the ACN campaign. Showing here, this is a sulfate. Again, the red is summer, the blue is winter. We see the higher sulfate concentration at a lower altitude. This is very consistent with the picture that the sulfate is mainly produced either there's ocean sources, ocean surface, the source from ocean surface. And the black carbon has a very different profile. We see there's a higher concentration in the lower free troposphere, about 2,000 meters. You also probably will notice that the average concentration is substantially higher than the median value. This indicates that there were occasional abnormal high concentration. This is attributed to the periodic transport of continental emissions, which have a high concentration of black carbon. And this is also supported by increased organic concentration and also back trajectory analysis, which I'm not showing here. So if you compare the vertical profile organics and black carbon, you can see the enhancement of organics during the summertime in the marine boundary layer cannot be attributed to the entrainment from free troposphere. And this is attributed to the surface sources such as the primary marine aerosols and also potentially secondary organic aerosols formed from oceanic VOCs, which I will talk a little bit later. So now let's look at the particle number concentration. We are interested in the CCM population. So I'm using the accumulation mode concentration as a sort of the surrogate of the CCM population. And you can see that we have enhanced the CCM concentration, higher CCM concentration during the summer in the lower free troposphere. Again, this is due to the more frequent transport, long-range transport of pollutions from, for example, from North America. Just like the black carbon, enhanced black carbon organic concentration I showed in the previous slide. But if you look at, compare the concentration in the boundary layer and lower free troposphere, you will find that actually the accumulation mode concentration in the marine boundary layer is higher than the free troposphere. So if you entrain the free troposphere air into the marine boundary layer, they actually do not directly increase the CCM population. On the other hand, the free troposphere has much higher Aitken mode particle number concentration. So the entrainment will increase the concentration of the Aitken mode particles in the marine boundary layer where they can grow and reach CCM size. So obviously, this represents a sort of like indirect sources of the marine boundary CCM because they have to grow to larger size. The strength of this source depends on the concentration difference in the Aitken mode between the free troposphere and the boundary layer, and also how quickly those particles can grow and reach a sufficient large size to serve as CCM. So we are particularly interested in what are the species we're contributing to this growth process, which represent a potentially important source of the CCM. So because Aitken mode particles are small in size, they have very little mass, it's actually quite challenged to, I mean, it's possible, but it's actually quite challenged to measure their composition with a high time resolution directly. So what we did is we take advantage of the hygroscopicity measurement to infer the composition of those condensing species. What we did is we first derive the particle hygroscopicity based on the size resolved CCM measurements. Essentially, we size select the particle and see under what kinds of supersaturation they will form, cloud droplets. That will provide us what is the hygroscopicity of those particles. Once we have particle hygroscopicity, we can derive the hygroscopicity of a condensing species by tracking the variation of the particle hygroscopicity during the growth of 8 chemical particles. Essentially, we follow how the hygroscopicity of the growing 8 chemical particles change as a function of time. This will allow us to derive what are the hygroscopicity of the condensing species. And based on the hygroscopicity of the condensing species, we can then constrain the composition by taking advantage of the very different hygroscopicity of the typical compounds, inorganic, organic. Basically, the organics typically have much lower hygroscopicity compared to the sulfate inorganics for condensation growth. For example, in this case, we can derive the hygroscopicity of the condensing species about 0.7. So this is likely dominated by condensation of sulfate. For this case, the condensation is dominated by organic compounds. So now based on the kappa of condensation species, we classify the growth events into 3 different categories. One that is more dominated by organic compounds. This include MSA, and also those are dominated by sulfate. This is larger than 0.65 when the kappa of condensation species is larger than 0.65. And if the value is somewhere in the middle, that would indicate a mixture of both type of species. So this shows the distribution of the 3 different type of the condensation growth events as a function of the months of the year. And you can see the blue represent those are dominated by organics. The brownish color represent that is a contribution from both organics and inorganic compounds. And organics represent quite important, or even the dominant condensation speed doing 80% of the growth events at the ENA site where we observe those events. So we don't really have any information about the precursors of those condensing species, but the earlier studies shows that VOC produced from marine biological activities and the VOC formed by oxidation at the air-sea interface could potentially generate VOC that can lead to secondary organic aerosol formation that contributing to this particle growth. We're also quite excited. We will be going to Azores again in less than 1 month to participate in another field campaign, which should help us to better understand the composition of those condensation species and also their precursors. So I want to also show quickly some of the earlier work we have done. This is the work done about a few years back. And if you look at the middle column, basically this shows the contribution of different processes to the accumulation mode or CCM population in the marine boundary layer. So except during the winter time, this is actually very consistent with what Trish have shown. We see that the condensation growth indicated by the yellow color here represent actually quite an important process for essentially the condensation growth of 8 chemical particles represent actually an important source of CCM in the marine boundary layer. And particularly in the summertime or in the fall time. And I also want to point out this analysis is based on the assumption that the condensation is dominated by the sulfate produced from oxidation with EMS without considering of any organic compounds, and therefore they likely represent a lower limit. So we know the condensation growth by organics in the marine boundary layer is often inadequately represented in many global models. So their contribution of the particle growth from 8 chemol to the CCN size to the population of the CCN is likely underestimated. So with that, I'd like to summarize. We see new particle formation occurs actually quite regularly under specific conditions. This is following the passage of a cold front in the upper part of remote marine boundary layer. We found that the high accumulation mode concentration in the lower free troposphere during summer is to a large degree due to the more frequent long-range transport of continental emissions from North America. We found the entrainment of free troposphere does not really directly increase the CCM population. Instead, it increases the 8 chemol particle number concentration, which can then grow to reach CCM size. And the secondary organic compounds represent an important or sometimes even a dominant condensation species during many of the growth events observed at the ENA site. And if that applies to other regions of the ocean, then the condensation growth of the nucleation 8 mode particles, the contribution to the CCM population may be underestimated in the global models. So with that, I'd like to first thank the group members who did actually essentially all the work, Zheng Guangjie, Wang Yang, Zhang Jiaoshi. And also, whenever you go to a field campaign, it's a big team effort. I want to thank the ACNA team and also the NAMES team, particularly the Las Atlantic group who did the aircraft measurements. And I also like to thank the funding agency from Department of Energy, our ARM program, and also Atmosphere System Research Program, the research aircraft, the pilots and the crew, and the facilities at the Department of Energy, ARM Climate Research Facility, Environmental Molecular Science Laboratory, EMSO. Last but not least, the government of Azores, who allow the aircraft to fly and to sample in the Azores region. So with that, I'd like to thank you for your attention. I'll be happy to answer any questions if there's time.
Thank you, Dr. Wang. We still have some time for questions. Is there any question from the audience? Okay, we have one question in the chat box. Are the iodine species contributing to the new particle formation in the ENA site? The iodine species.
Okay, no, I don't think so. We don't think this is new particle formation due to the iodine emission, for example, along the coastal area. First, we see that more originally occurs in the upper part of marine boundary area. In addition, we also try to correlate what we observed on the ground. Essentially, we see the aircraft only tells you, we have limited, aircraft only have 2 seasons. We observe those newly formed particles also from the ground measurement, which is more long-term. We try to correlate the time when we observe them. They're not really happens during the low tide. So typically when you see the iodine, you would see there's a low tide, you expose the algae along the shorelines, you have the iodine emissions. We don't really see there's a correlation between there. So we're pretty confident that those are not due to the iodine species.
Okay, the next question is, you mentioned the upcoming campaign. I'm interested in knowing what VOC species or organic species will be targeted for the linkage with new particle formation.
We don't know for sure. I think maybe, I don't know whether Nate will talk more about this in the phone, but essentially there will be PTRMS measurements. We'll be trying to measure the Euro suspects, maybe the isoprenes or the monoterpenes. And I know those emissions are typically very low. There are some studies showing they're very isolated time. There were higher emissions. We don't really have, I mean, there are also papers published about oxidation of the oxidation layer of the ocean surface that could produce the VOCs. We don't really have a clear idea, so that's actually part of make it a little bit exciting, that we will try to see what we can find out. Yeah.
Okay. Actually, I have a question about the new particle formation and when the temperature change. So what about the temperature dependence for this new particle formation, especially when you have the organics, right? And the phase state of organics is quite depend on the temperature and the related humidity. I guess Nate will talk about that more later. So when the cold front reach to the ENA site, and how does this new particle formation react to this temperature change? So response to the temperature change.
Right. So I think probably the main reason of why you would have new particle formation after cold front, I think the cold temperature is one reason, but I'm speculating here. Of course, we have not completely simulate that events yet. So we don't really quite understand the mechanism at this point. But I would say probably the most important factor is probably the surface area and also the DMS concentration availability. This is just like the conventional view that you have this new particle formation in the outflow of convective clouds. Essentially, the precipitation removes the existing surface area. So you create a condition that is really clean between those clouds. The cold temperature is, I think it's a favorable condition, is that because if given everything else equal, because when you lower the temperature, essentially reduce the volatility of any participating species. That's essentially accelerating new particle formation, right? Because given, let's say everything else equal, you're producing the same amount of, you have the same concentration of a nucleating species, lower temperature just reduce the volatility of the nucleating species. But I think the temperature is probably a little bit more complex parameter there, because when you have a different temperature, that may also affect the reaction rates, which could affect the nucleating species, the concentration of nucleating species too. So what I'm trying to say is I don't really have a good answer. I think it's a little bit, we are trying to, in an effort, try to see whether we can model those nucleation events using the chemical mechanism that is observed in the laboratory, see whether we can really sort of have a closure. We can really predict those kinds of new particles observed. We have a project we are starting. We haven't done it yet, so I don't really know the answer at this point. Yeah.
Okay, thank you. Trish has a question. Can you say again what fraction of new particle formation is happening in the upper marine boundary layer versus free troposphere?
Right, so that's very good. So of course, I have to say, this is all based on limited data. So essentially this is based on the 3 names field campaign data. And we found this is about 6%. Essentially we take the measurement. Of course, we're making assumption the measurement is more or less random, right? Essentially we are not, essentially the aircraft measurement is more random. We take all the time when we see new particles, and then we divide by the total sampling time for that altitude range. And we see here, there's a 6%. And in the free troposphere, we have 15%, right? I mean, this is in the altitude of convective clouds outflow. My feeling is, of course, if you look at the number of particles being produced, it's much more. But this has an advantage. First, is closer to the clouds, right? Because it's already in the marine boundary layer. If they can really quickly grow, that could have a substantial contribution. Of course, that is sort of a speculation at this point too. We are trying to simulate this. We still have not done it because it's actually quite challenged to do a convincing job. I hope that answers your question, Trish.
Okay, thank you, Dr. Wang. Could you stop the sharing? And,
yes.
Let's move to the next speaker, Jonathan Slade. Nate, could you please share the screen or the slides? Yeah. So Nate is William Lee Assistant Professor of Chemistry at the UCSD starting from 2018. He got his PhD degree at Stony Brook in 2015, and he was a member of the atmospheric chemistry curriculum for emerging senior scientists, SS. So Nate will bring us a talk on molecular drivers of nascent and aged sea salt particles phase states across the phytoplankton bloom. Nate, please.
Right, thank you. Hopefully you can see my full screen.
Yes, we
Okay,
can.
great. First, want to again thank the organizers, Drs. Wang and Lee, for the kind invitation to present today. And what a great set of talks so far. It's helping in terms of providing background for what I'll be talking about. I guess a little bit of background about me. I work closely with the National Science Foundation funded Center for Aerosol Impacts on Chemistry of the Environment, or CASE, at University of California, San Diego, and Scripps Institution of Oceanography, whose main motivation is to understand the chemical and physical processes involved in the formation and transformations of marine aerosol particles. And my research group's contribution to CASE objectives so far has been to understand chemical interactions in the particle phase of sea spray and how they influence physicochemical properties of sea spray aerosol, including viscosity or phase state on a more molecular level, especially across the phytoplankton blooms, looking at how differences in biological activity in ocean water influences the composition and phase state of sea spray aerosol. So before I get into my talk, I'll acknowledge those up front who contributed to this work, my grad students, Ryan Tuminello, Adam Cooper, and Samantha Cruz, and undergrad researchers, Karen Lopez-Zepeda and Reese James. And this work was mostly funded by CASE, as well as some support from the Graduate Research Fellowship Program from the NSF for one of my students. So again, Trish and Jan gave a great introduction to sea spray aerosol. I'll just briefly go over it. It's the most abundant aerosol in the atmosphere by mass. It's created when waves break, when bubbles form at the surface and rupture, sending out the residual non-volatile material into the atmosphere that can be lofted to higher altitudes and transported over long distances. Those bubbles can scavenge mostly surfactant-like organic molecules, short and long chain fatty acids at the surface layer, the sea surface microlayer, as well as send out more water-soluble organic compounds like saccharides and hygroscopic inorganic salts and biological material. So these the chemicals in sea spray aerosol can undergo different interactions that influences not only its composition, but its physicochemical properties, including phase state. Now, sea spray aerosol, as mentioned previously, is in terms of its impact on climate, is highly uncertain. And part of that uncertainty, I think, also falls under a lack of detailed understanding of its molecular composition and transformations in the particle phase, and also those impacts on its physicochemical properties, including their role in ice nucleation and cloud condensation nuclei formation. So I'm focusing on viscosity. Viscosity we define as the degree of frictional forces in materials and the strength of intermolecular bonds. It influences things like molecular diffusion in particles. So how into the particle bulk will gases diffuse into the particles, or how organic and other materials in particles will diffuse to the surface and undergo reactions at the surface. It controls those kind of processes kinetically. Surprisingly, there's been very little research on the phase state of sea spray aerosol. However, I will highlight one study that was published in 2020 by Lee et al. From the Tavansky lab at University of Iowa. In collaboration with CASE, they sampled or collected sea spray aerosol generated in a wave flume mesocosm and analyzed its properties on a single particle level using an atomic force microscope. And what they observed during that study was that during periods of high biological activity, the organic portion of sea spray aerosol became more semi-solid-like, and they attribute that to an increase in the organic volume fraction. However, what's really lacking in this study and others, I think, is a lack of a detailed understanding of what's really driving those differences on a molecular level. And so those are two questions we want to ask is how does sea spray aerosol viscosity change as a function of biological activity in clean background air? So this is in the absence of photochemical oxidants. So this is the primary portion of sea spray aerosol without influences of polluted air. And following up with that, what are the driving molecular properties controlling sea spray aerosol viscosity changes? So we needed a way, in order to do this successfully, we need a way to generate sea spray aerosol particles with the proper size distribution and composition that you would find in coastal marine environments. And so CASE has worked very hard over the last decade or so to bring the ocean into the lab, where we take bulk ocean water samples off of the Scripps Pier at SIO, and we place that into a long wave flume mesocosm. Waves are generated mechanically with a paddle and then break on an artificial beach. And that generates sea spray aerosol particles, as well as a bubble size distribution in the water that's consistent with what's been observed in field studies. So we're able to generate sea spray in a controlled way. And we participated in this study called Sea Spray Chemistry and Particle Evolution Experiment in 2019, where our objective was to sample these particles and analyze their phase state and composition. So this is a large study, and our setup is shown illustrated on the right. I'm not going to go through and bore you with all the details, but the key takeaway here is we sampled the headspace of this wave flume mesocosm, and we either sampled the nascent portion, or we passed those particles through an oxidation flow reactor to study aging effects by hydroxyl radical oxidation. We then controlled relative humidity downstream to assess changes in how relative humidity and aerosol liquid water content will influence the phase state of sea spray aerosol. And then we had a variety of measurements downstream to analyze the physical and chemical properties of these particles. And so measuring viscosity is a challenge, especially for submicron sized particles. In sea spray aerosol, the dominant number weighted diameter is approximately 200 nanometers. So far, there are no techniques that exist that can measure that viscosity directly. So we take an indirect approach using well-vetted methods published in the literature to assess the phase state of these aerosols based on particle bounce fraction measurements using an electrical low pressure impactor. Now, the basic operating principle of this method is we sample the aerosols and we accelerate them towards a rigid surface, a smooth, rigid surface. And we measure or quantify how many particles we sample and how many bounce off of that surface. So you can imagine for a more viscous or solid-like particle, we'd have more of them that impact and rebound or bounce off of that surface, whereas liquid-like particles will tend to stick at that surface upon impaction. In order to get to molecular drivers, of course, we need compositional measurements, and my group deployed an extractive electrospray ionization mass spectrometer during that study to analyze the chemical properties of the organic components in sea spray aerosol. Just to illustrate this process quickly, basically we're sampling particles online, aerosol online. We're not impacting on a filter first, for example, and extracting it. We expose the aerosol sample to a plume of electrospray droplets consisting of either acetate ions in negative mode or sodium ions in positive mode. The coalesced droplets undergo chemical ionization in the droplet phase, and in positive mode that typically forms sodium adducts with those organic compounds. And that's sent to our mass spectrometer. So because this is a high resolution method, we're able to obtain with some pretty good accuracy the molar, we can predict molar mass based on their chemical formulas, as well as oxygen to carbon ratios or carbon oxidation state. This is a huge collaborative study, so we were very fortunate to work with other groups who had an aerosol mass spectrometer where we were able to see or measure organic and inorganic mass fractions of these aerosol particles, as well as the hygroscopicity parameter or hygroscopicity, the affinity of these particles for water, using a size resolved cloud condensation nuclei counter. So we took these two sort of separate approaches and we combined this information into a theoretical model to estimate viscosity, particularly of the organic material in sea spray aerosol. And this is based on formulations that have been well described in the literature over the last 5 or 6 years. Apologies, I have some background noise. Okay, so I want to first walk you through our first observations during the study of the bounce fraction as a function of date, which is over the course of a phytoplankton bloom. The bounce factor values are shown in the white box plots. The red bar in those box plots correspond to median values, for example. And these are all binned daily. So we sampled overnight in the absence of solar and artificial light over the course of about 6 hours. And so each of these box plots contain at least 60 independent bounce fraction measurements. And so we bin them daily. Chlorophyll A, which is a proxy for phytoplankton productivity, is shown in the green, and bacterial cell counts are shown in the gray and correspond to the right y-axis. And then we've just, I'm showing here this dashed red line, these dashed red lines, which shows typical separation in phase for liquid, semi-solid, and solid particles based on previously measured bounce fractions of these different types of particles. And so to get into some of these results, what we observed during periods of low biological activity in the study, or what we call the pre-bloom in this study, was that the particles were consistently in what we would consider a liquid-like phase state with a bounce fraction less than 0.2, meaning that less than 20% of the aerosol population we sampled bounced upon impaction. However, as the biological activity increased and the phytoplankton bloom kicked in, we saw the significant enhancement in the bounce fraction of these aerosol particles, which indicates the population of the aerosol became more viscous with median bounce factors on the order of 0.8. Now, this was a rapidly decaying bloom. It only peaked for the course of 1 or 2 days and then decayed rapidly. And we saw that corresponding to the decrease in chlorophyll A, that the bounce fraction also decreased into what we call the post-bloom. Now, to me, this was sort of, it was an astounding finding because this implies that we can look at chlorophyll A as maybe an indicator of the phase state of sea spray aerosol. The higher the biological activity, the more viscous the aerosol. The lower the biological activity, the less viscous. Now, this was for one experiment. I'm not showing you the other data because we don't have enough time, but we did do 2 independent mesocosm studies before and after this in my lab on a smaller scale, induced a phytoplankton bloom and measured the bounce fraction of those sea spray aerosol. And it showed very good correlation with chlorophyll A content in the seawater samples. But we can't stop there, right? So we need to know what is driving this difference in phase state from these different stages of the bloom. So what we're showing here is the bin data for the calculated oxygen to carbon ratios in the white box plots and the molar mass of the particles that we could determine based on our mass spectrometer measurements in the blue box plots. The takeaway here is that during the pre-bloom stage, we found that consistently that the median molar mass was about 150 grams per mole less than what we observed during the peak bloom and into the start of the post-bloom. Now, this is an important metric for phase state or viscosity because as you increase molar mass, you increase the size of the molecules in the particle phase, and thus the probability that these molecules interact with other molecules and cause frictional interactions. And that's what we define as viscosity. But there's another player here, liquid water. So water acts as a plasticizer, and so we need to know what contribution does liquid water have to the phase state of these aerosol particles over the same period. So the CCN measurements of the hygrosphericity parameter allowed us to derive the hygrosphericity parameter of the organic components in sea spray aerosol. And that's shown in the red across the bloom. The white box plots indicate the organic mass fraction as measured by the aerosol mass spectrometer. And what we observed was that during the peak bloom period, there was a couple of days here we see where it's less, the hygrosphericity parameter is less than what we see for the pre-bloom and post-bloom. So this indicates that the organic components in the sea spray aerosol are becoming less hygroscopic during the period of peak biological activity, and implying there's less liquid water in the aerosol phase and potentially influencing that increase in the bounce fraction that we observed. So in order to estimate viscosity, we had to calculate the glass transition temperature of the organic molecules following previous methods. And what I'm just going to define really briefly, the glass transition temperature is a range at which a material transitions from a semi-solid phase state to a glassy-like phase state upon cooling. And this is directly related to its composition in terms of molecular weight, oxygen to carbon ratios, and the hygroscophicity parameter, as well as the fraction of the particle that is organic. And we used that and calculated the viscosity based on Stokes-Einstein relationship. So I'm going to bring you back to what the bounce fraction looked like in this plot. And now we're going to see how the calculated viscosity changed over the course of this bloom. And so what we observed is, let me describe it first. In blue is the viscosity of the organic material with the influence of liquid water. The black trace, that indicates the mixed particle composed of sodium chloride viscosity at its given water activity and the viscosity of the organic material. Let's focus on the blue for a moment. The blue trace, we see that during the pre-bloom period, we predict based on our compositional measurements and hygroscophicity that the particle viscosity is on the order of that of ketchup or honey. Whereas if once you transition to the peak bloom period, the viscosity enhances to something similar to peanut butter or caulking material and then decreases into the post-bloom, similar to what we observed in the measured bounce fraction. Now, if we assume the particles are mixed, we can also calculate the viscosity of those with sodium chloride, and we would predict that these particles are always in a liquid-like state, which is not what which is counter to what we observe from the bounce fraction. And this is likely due to the assumptions of that calculation, which is it assumes that the particles are homogeneously and well mixed. But we know from observations in the field and in previous studies with this wave flume mesocosm that sea spray aerosol can exhibit different morphologies and phase separation. So they're not necessarily homogeneously mixed. There's a lot of work that still needs to be done to relate phase separated particles with homogeneous particles to viscosity and bounce fraction. So why do we care about this? What are the implications? One way we approached this was to take all of our compositional data and based on typical vertical profiles of relative humidity and temperature in a simulated marine boundary layer in the absence of or in under cloud-free conditions, we calculated or estimated the viscosity of those particles during these 3 different periods of the bloom. Now, what we observe is that typically, because of the high relative humidity in the marine boundary layer, the particles are going to exist as liquids around a kilometer, so up to about a kilometer. But once we get above that range, under these conditions of temperature and relative humidity, we see that there is separation, right, between peak bloom and the other periods. That is, the particles during the peak bloom phase are more viscous at lower altitudes. So that could be important for someone who might be interested in ice nucleation, as more viscous organic type aerosol particles have been shown to act as better depositional ice nuclei. And so we're showing that this can happen or occur at lower altitudes. But marine aerosol is not limited to sea spray. In the real ambient environment, we have photochemistry. So reactions, gas phase reactions with VOCs, for example, DMS and other biogenic type VOCs can produce secondary organic aerosol and secondary organic material that can condense onto pre-existing aerosol. We also have heterogeneous interactions between photochemical oxidants and the nascent sea spray aerosol and the other particles that are present. So it's very heterogeneous in the real ambient environment. What we just showed was a very sort of simplified case of just the nascent sea spray aerosol portion. So we asked this question: how does oxidative aging of nascent sea spray aerosol in the presence of VOCs affect the marine aerosol phase state? So under conditions that we might expect in, let's say, a polluted coastal marine environment. So the top panel here shows the submicron volume size distributions for the different stages of the bloom, where the red indicates the volume distribution of the nascent sea spray aerosol, and the blue represents the submicron volume distribution of the aerosol after 7 days of equivalent aging by hydroxyl radical in the atmosphere. On the bottom plot, we're seeing the bounce factor for the nascent portion, which we saw before for these different stages, along with the bounce factor for when we age these particles over the course of 7 days. Now, there are important things to point out here. We had a lot of VOCs being oxidized with high concentrations of hydroxyl radicals. So we're producing a lot of secondary marine aerosol. You can see the peak in this volume distribution in the blue is somewhere around 20 to 40 nanometers, along with nascent sea spray aerosol. And we also saw evidence of condensation of these vapors on oxidized vapors onto pre-existing aerosol, as noted by this increase in the volume of these larger accumulation mode size sea spray aerosol particles. And that was also observed in the post-bloom. So we have this very heterogeneous mixture of aerosol. And it's really difficult to pull from that, which one of these is really contributing to the phase of the aerosol that we're measuring. But it goes to show that once we start aging this mixture of VOCs and nascent sea spray aerosol, that the majority, at least based on number concentration, of the particles that end up bouncing are larger after aging of 7 days. During the pre-bloom and peak bloom stage, we see that increase from 0.2 to about 0.8. Whereas in the peak bloom, there was very little differences between the two. And we attribute this to differences in potentially water solubility, but we see differences in the calculated hygrosophicity parameter of the organic material. After aging, those particles become less hygroscopic from what we've observed. During the peak bloom, we see there's very similar hygrosophicity parameter, which is consistent with our observed similarities in the bounce fraction during that period. We also age these particles out to atmospherically irrelevant timescales, typically 14 days to 24 days, as shown here. And what this indicates is that with extensive aging, that the particles become more liquid-like. And we attribute that to what we've observed in the literature before, which is that high high degrees of oxidation by hydroxyl radical that can begin to fragment and volatilize organic components on aerosols, baking them off essentially. And what that leaves behind is the inorganic salt, which is more hygroscopic, potentially leading to more liquid-like phase states after extensive aging. Because this was so complicated of a system, we wanted to try to better understand, based on one interaction, one aging type through heterogeneous oxidation in the absence of VOCs, how does that contribute to observed phase changes? And so we've been making sea spray aerosol mimics in my research lab, starting out with a binary system, just simple systems using saturated fatty acids as representative organic molecules, including lauric acid, a C12 fatty acid, and sodium chloride mixed with sodium chloride. And we varied exposure in an oxidation flow reactor up to about 5 days of equivalent aging for the pure lauric acid aerosol particles and lauric acid mixed with sodium chloride to observe how oxidation affects the phase state of those aerosol particles. And what I'm showing here is the relative volume change in the top panel and the bounce factor on the bottom panel as a function of aging in terms of equivalent days in the atmosphere. And so what we observed in the red is for the pure lauric acid particles, that we lose about 80% of the volume as a function as we age it over 5 days. Whereas the mixture with sodium chloride, we only see about a 20% decrease. In contrast with the bounce factor, we see an increase in bounce with the lauric acid particles, whereas the mixture with sodium chloride, we observe no significant change with increasing aging. So what is driving these differences between these two aerosol systems? One which is highly organic, the other one which is mixed with an inorganic salt. So we can glean some information from earlier works showing molecular dynamic simulations of fatty acid in different protonated, different pHs with different concentrations of sodium ions in the particles. And what they show is that for fatty acids in a protonated state, which is what we'd expect for lauric acid under higher acidity conditions, that they form more rigid spherical structures. But as you increase the amount of sodium ions in the particles, this begins to deprotonate the carboxyl head group of these fatty acids at higher pH. And what that ends up doing is deforming this spherical structure into this more deformed structure, and that allows more migration of sodium ions and liquid water throughout the particles. So that can explain perhaps why we see these kind of differences as well as the hygroscopicity of the lauric acid versus lauric acid plus sodium chloride. But what happens with aging? Why do we see an increase in bounce with increasing aging? Well, we know that hydroxyl radical oxidation of saturated fatty acids is primarily going to attack one of the hydrogens on the CH2, abstracting a hydrogen, and that can undergo different functionalization pathways as well as decomposition to form aldehydes. Because we observed significant volume loss of the organic components of lauric acid, we suspect that most of that is undergoing the decomposition pathway and forming these aldehydes. And previous studies have shown that when you start to volatilize or evaporate a particle, under these acidic conditions in the presence of aldehydes, we suspect or hypothesize that we're getting potentially aldol condensation reactions or accretion reactions under these more acidic conditions, where we can start to form higher molecular weight and more oxygenated beta hydroxy and alpha beta unsaturated aldehydes, for example. Now, there's still a lot more work to do with this. We're in the process of examining the mass spectra to see, do we observe these kind of reaction products? And we're also exploring how different inorganic salts like calcium chloride and magnesium chloride, which have a different charge state than sodium chloride, can impact the phase state of these aerosol particles. Some previous work has shown that divalent cations, for example, are better at forming organic cation interactions in stronger bonds that could then promote more viscous phase states of these aerosol particles. I don't have a conclusion or summary slide. I just want to thank my group. Those highlighted here contributed to this project, and the entire Seascape team, especially Kim Prather, one of the directors of CASE, and the other team members for making this project happen. Thank you.
Thank you, Nate. So I do have a question about the size dependence of phase state. So when you do the lauric acid oxidation, you see the size change, decrease the size. And how does that response or how the bounce factor responds to that?
Yeah, it's a good question. So you know, with this bounce method, I didn't mention it before, but we're not doing size resolved bounce fraction measurements. So we're just taking the population of the aerosol. Now for this experiment, these are in the lab, we control them. So we do send in a size resolved around 100 nanometer diameter particles for this study. But I would suspect that, you know, as you start to, you know, lose that organic material and form smaller particles, it really depends. If it's smaller particles, you might think they're less dense. They'd have less momentum upon impaction, potentially leading to more liquid-like type phase states. But since we don't see that happening, you know, I think we're, you know, it may not be necessarily a size dependence on bounce here, but more how the composition of those aerosol particles are changing. And I will note that how we calculate the bounce measurement is we do account for the size. So we account for the signal of the particles that bounce at a given size with what we measure in the absence of bounce, if that makes any sense. That's how we derive our bounce fraction measure.
Mm-hmm. Okay, thank you. Okay. I was logged out, so I can see the screen. This is questions with the format. So what is the relative humidity when measuring the bounce factor? Is the size resolved bounce factor or batch mode?
Yeah, that's a great question. So it depends on which experiment. During the Seascape experiment, we adjusted relative humidity, and what the results that I showed were chosen for 50% relative humidity. just sort of representing more transported over land type of sea spray aerosol particles. In this particular experiment, these were done at, let's see, so yeah, these were first passed through a diffusion dryer, so this dehydrated the particles. And then we, but it doesn't mean they effloresced. And then we increased the relative humidity where they were oxidized in the oxidation flow reactor, and that was around 50% as well.
So in your mesocosm study, the phytoplankton bloom. So you mentioned that the correlation between the chlorophyll A and the bounce factor. So, but to my understanding that the chlorophyll A somehow represent the part of the organic material, maybe. But what do you think about that? Why is that so strong correlation that you mentioned, or it's just one or two cases?
Yeah, no, it's really, I think it's challenging to answer. We provided some hypotheses in the paper that was published in 2021 on this. We think that we think there is some biological mechanism that's in play, right? For example, I think during the pre-bloom stage, and there's a low biological activity, which is sort of homeostasis. You have smaller molecular weight sugars, glucose, for example. But as you start to increase that, the formation of phytoplankton and other biological material and microbes, then you're getting organic compounds that are associated with cell wall formation. You're getting these longer chain fatty acids, lipopolysaccharides, larger molecular weight molecules. So I wouldn't say there's a direct mechanism for chlorophyll A in terms of its impact on the phase, but more indirectly, what it means when chlorophyll A changes in the ocean. You're going to have different chemicals at play that lead to the formation and decomposition of that phytoplankton.
Okay, so now let's see. Do we have other questions from the audience? To Nate? Yeah.
I have a quick question, if I can ask.
Please go ahead. Yeah.
Yeah. So Nate, you're showing this this time series pre-bloom and bloom, and if I remember correctly, there was actually one data point that shows actually you have a lower organic fractions during the.
Yes.
Was that is that right, or do you have any?
Yeah. So this figure, right. There's 2 days, August 2nd and August 4th, which showed these relatively lower organic mass fractions, which was also pretty surprising to us. I guess I should be careful here because I know the group that made these measurements insisted that, well, we got to be careful with this one because these are kind of, the organic mass was so low those particular days that they're not confident in the quantification. But just the fact that they're low means that there's less than what there was in the pre-bloom and post-bloom stages. And it is kind of contradictory to what I would expect if you have more organic volume, as maybe is proposed by the Lee et al. Study, that you'd have a greater organic mass fraction. And that I think is really, that's driving that decrease in the hygroscopicity when you convert this to a volume fraction and you calculate kappa of the organic material based on that volume fraction. Yeah, it's interesting because while the hygroscopicity parameter of the SSA by itself remained approximately constant, somewhere around 1.1 to 1.2.
Yeah.
The inorganic salt, we assume, is sodium chloride here, and we take it as 1.5 as the kappa value. And that, because of the lower organic mass fraction, that would mean that the hygroscopicity of the organic material is less during those periods.
Yeah, I also was wondering, because there are quite a number of studies shows that you have relatively high organic hygroscopicities. Even for there's basically there is some kind of differences in hygroscopicity under the supersaturated conditions and undersaturated conditions, right? So organics in our certain studies shows even you have a lot of organic, it doesn't really change the kappa for CCA activation. Yeah. So if that's the case for apply here, that would mean you would also derive a relatively high hygroscopicity for organic compounds, right? I mean, because even you add or if let's say you replace sulfate with organic, you don't really not sulfate. You replace the sea salt with organic, it doesn't really change the hygroscopicity as much. So essentially, the organic would have derived organic kappa would have relatively high value as well, right? So
yeah.
I thought you would probably one better kappa value to use is from hygroscopicity growth instead of CC activation.
Right. Yeah.
This type of
and yeah, it's challenging. Yeah, we're trying to understand what could be driving that difference in the organic kappa value. There has been some literature on I've never seen organic compounds with a kappa value of 0.7 or large, but they're larger, right? But there are some compounds like glycine, which has a kappa value of somewhere around 0.7. But also this could be influenced by surface tension changes.
Yeah. And liquid phase separation. There's a number of theories have been put forward to explain the relatively high hygroscopicity, even you have a large fraction of organic compounds.
Right.
But it's very interesting. Yeah, it's very interesting results. And yeah.
Thank you.
Okay. Yeah. Thank you. Thank you, Nate, for talk. So it's already 11. So yeah, before we close the section, I would like to say something that maybe. So we are fortunate to have 3 great talks today and showing us the recent findings obtained from the lab and the field measurement and on the marine aerosol physical and chemical properties. And then we have discussed the links among the phytoplankton blooms and aerosol processes with the aerosol-cloud formation properties. So I would like to ask the speakers a general question. So about your opinions on, so what are the key questions that remain we may need to address or to answer for better understanding in these connections between the bloom aerosol processes and their cloud formation, especially when we have this new oxidation pathway as Trisha mentioned. And then we have this unknown organic VOC oxidation contribute to the maybe the new particle formation and to the CCN concentrations. And Gen 1 also talked about the seasonal and synoptic conditions that will also affect the new particle formation and then to the CCN. So with the new findings in the last like 10 years for the phase state of organic material, both for the SOA or primary or organic material. So could you give us a quick short comment on this? Nate, can you start that?
Oh no, this is hard to answer. I'm relatively new to the marine aerosol field. Yeah, I mean, I think as what's been going on here with trying to deconvolve, the portion that is from oxidation of VOCs, in terms of their impacts on CCN, and separating that out from the sea spray aerosol portion is really important. Being able to predict these kind of things based on simple, more simplified approaches may make it easier, more palatable to the community. For example, just looking at the phytoplankton growth as a metric. That's my start, but I don't know if that satisfies your question, Bingbing. It's a great one though.
Yeah, that's okay. I understand that. Yeah. Okay, thank you. Dr. Wang, do you want to say something on that?
Well, that's a really hard question. I'd maybe just say some of the puzzles that we're trying to understand. I mean, first, I sort of agree with that if you look at the new particle formation, we see there's sometimes it happens in the boundary layer. We see that actually happen quite regularly, but we don't really know how important that is. I mean, my feeling is still the free troposphere new particle formation probably is most likely still the major one, but the new particle formation in the boundary layer could be substantial. Has a potential, could be substantial. We don't really know, so that's something we try and find out. We don't even know at this point what the mechanism could be. We did some simple calculations based on the cloud chamber experiments, see what are the potential pathways. It sounds like you don't really need a lot of monoterpenes to produce organic compounds such that you can greatly accelerate the new particle formation rate. But that doesn't mean if we put those in, we can simulate it. We are still trying to simulate to see whether we can really using those mechanisms to reproduce. So we don't know. There are some evidence that both from laboratory experiments. I think this is from the Casey. How do you say your center? Case Center.
Case
Center. Yeah. So, and also from, there seems to be indicating that there's some evidence that the SOA could help the particle to grow. I don't really know how important that is. At least, there's a reason to believe that could be potentially important, could be not. So those are the some of the questions I'm thinking about, and also we have tried to do some preliminary comparisons with global models, right? Let's say, when do you say you understand this? That actually models can really reproduce what you observe. I think the models still, they may potentially produce. For example, I know at the Azor side, we did a preliminary work that is the CCM population may not be produced very well, certainly not at all supersaturations, right? I mean, there's a range of supersaturation, not all of them are produced well. So it's one location, but Azores are pretty representative of a much larger area. So that gives you thinking what's going on with the model, right? The model may need some improvements. So I don't really have a great answer, but those are some of the questions we're thinking about and try to figure out to get some answers. Yeah.
Okay, thank you, Dr. Wang. Dr. Green, do you want to add additional comments?
I think I can't offer more answers, but I can certainly say what I think the biggest questions are to date. And I think that this new oxidation product, HPMTF, is kind of given a whole shift on our thoughts of DMS and sulfate production. And I mean, it's irreversibly lost in clouds, meaning that maybe it takes away some of the importance of sulfate in producing new particles, and maybe it's more sulfate is adding mass to particles, moving them into the CCN size range. So that's an area for exploration. I really appreciate what Jay Wang was saying about the role of this upper marine boundary layer new particle formation versus the free troposphere particle formation. I think that's a huge question, because as he mentioned, I mean, if you're making new particles in the upper boundary layer, that's a much more direct connect to CCN and clouds in that region. And then the next big question that this discussion brought to me was the role of VOCs and how significant are they really in marine emissions of VOCs in producing SOA. So
Yeah.
I think those are three big questions that younger scientists need to go out and figure out.
Okay, thank you, Dr. Payne. So do we have other comments from the audience online? Okay, so Lily has a comment, yeah.
Yes, thank you, all the speakers and the participants. And I'd like to mention that back in 2018, so SOLAS organized a workshop. So recruiting scientists participating in 11 field campaign that addressing at least part of the science of the ocean ecosystems, aerosols-cloud linkage. And actually Trish was there and many people were there. And I saw some people online were there as well. So the aim of that workshop is to let the scientists be aware of what each other is doing and reach out to others for cross-cutting research. And there was extensive discussion at the workshop, like how to ensure the comparability between measurements and results conducted during different campaigns, and the necessity to compile a table of measurements necessary to study the ocean ecosystem aerosol cloud linkage, something like that. And the participants of that workshops, they showed strong interest to stay connected and talk to each other. I mean, the world is restarting now, so SOLAS would be more than happy if the willing is still there. So that's my comments.
Okay, so we already 13 minutes late. So if we don't have any comments, I would like to, before closing, we would like to thank again the three speakers for the excellent talks. And also we would like to thank all the participants, especially those who stay overnight in Europe. And the seminar is recorded and may be posted on the SOLAS YouTube channel. So stay tuned for the next SOLAS seminar. So we now can close the seminar. Okay, thank you.
Thank you
Again,
very much.
Bye.
Thank you.
Bye.
Thank you, Dr. Wang and Dr. Li, for the invitation.
Okay, thank you.
Thank you for joining.
Bye.
Thank you all. Bye, Jan.
We need to talk.
You may wrap
Nate,
up.
are you?
No.